Welcome back. So in this lecture, I am going to finish up on the protein ligand interaction and I will talk about allostery of proteins. So I will complete the discussion on protein DNA interactions. So that will complete the discussion on protein ligand interactions. Then I will define what is allostery. As an example, I will discuss the structure of hemoglobin. I will also discuss models of cooperativity and what are the roles that pH and BPG play in modulation of oxygen binding to hemoglobin. So we have already seen this in the previous lecture slide that there are different types of protein ligand interactions. It can be protein small molecule interaction, it can be protein interaction like in case of hormone and cell receptors, or it can be protein DNA interaction. So we are going to focus mostly on this. Protein antigen interaction is also antibody antigen interaction is also protein interaction. So today I am going to focus on these interactions where a protein interacts or binds DNA specifically, right? So it can also bind DNA non-specifically, but we are going to talk about specific binding. And what I mean by specific binding is that the protein identifies a particular sequence of nucleotides on the DNA and binds that sequence. So these are the two types of protein ligand interactions that we have discussed. So I am going to finish up on this where a protein is there and it binds the ligand and the protein ligand complex is formed. And then we also discussed two types of modes of interaction. One is lock and key where the ligand fits perfectly to the binding site, or induced fit where the ligand loosely binds and then that results in a structural change in the protein and then the ligand binds more tightly. But both will come under this type of interaction. The second type of interaction which I am going to focus mostly today is allosteric interaction. In this case, there are two molecules. One molecule binds to a particular site of the protein and this type of molecule, here it is called ligand A, but we will call this as a modulator. So a modulator will bind here and that binding will result in the shape of the binding site of the protein which is at a distance. And then the protein will bind the ligand B, or which we will call ligand. So this is ligand, this is a modulator. So modulator binding results in a change in shape of the binding site for this ligand and then the ligand will bind. So this will be a positive modulator. We will also see negative modulator where ligand can bind, but when the modulator comes and binds, the ligand cannot bind. Right? So we will see examples of this. Okay, so let's get back to this non-allosteric interaction. So this is just a basic refresher on the structure of DNA. You have already seen this. So DNA is a double helix, has a double helix structure where two strands form complementary interactions with each other. So that's the backbone of the DNA and the bases are forming hydrogen bonds with each other. So we have AT and GC pairs inside. Now if you look at the structure of the DNA, even though it is helical, it is not symmetric. So what I mean by that is if we look at the distance between the two strands, it turns out that the distance are of two types. One is, so in this case you see the distance is 15 angstrom. So this creates a groove. So if we have this sphere, we represent each atom as a sphere instead of ball and stick, then you can clearly see a formation of a groove. You see this is a groove, right? And this groove is called the major groove. If we look at the other side, so this one or this one, which is shown here in more detail. So you see here the distance is smaller, it is 7.3 angstrom. And you can see that the two strands are very close to each other. The gap between them is much smaller. So this one is called a minor groove. So there is a major groove. So this will be the major groove and this will be the minor groove. In case of major groove, the distance between the two strands is 15 angstrom. In case of minor groove, it is 7.3 angstrom. So the distance is actually almost half, actually less than half than that of the major groove. Another interesting aspect of DNA structure is if we look at the backbone. So the backbone is formed of phosphate and sugar, right? So they are exactly the same for all the nucleotides. The difference comes from the base, nitrogenous base, which is either A, T, G or C, and those nitrogenous bases are here. Now if a DNA has to bind specifically, to a particular sequence, it has to make contacts with the bases. If it only interacts with the backbone, then the backbone is exactly the same for all nucleotides, right? So it has to somehow interact with these nitrogenous bases. And we will see that the majority of these interactions are either hydrogen bond or hydrophobic interactions. So if we focus at this AT and GC pair, so the GC pair, GC or CG is shown on the top, AT or TA is shown at the bottom. So if you focus on this structure, you can see that there is the sugar and then there is this nitrogenous base, that is your G. For the complementary strand, this is the sugar and then there is the nitrogenous base, which is C. So G and C form 3 hydrogen bonds like this. Similarly, you can have the reverse, which is C and G, right? This part of the base forms the major groove and this other side forms the minor groove. And if we look at the functional groups which are exposed that the side chains of proteins or amino acids on the proteins can interact with, then we will see these are the functional groups which are available in the major groove. So this is red, hydrogen bond acceptor. This is also hydrogen bond acceptor, which means that NH or OH type of groups from the amino acid side chains or even the backbone can interact with this. This is a hydrogen bond donor, so this can interact with oxygen or C double bond O kind of groups on the protein side chains or the backbone, right? This is just a hydrogen, so it can form some sort of hydrophobic interactions, right? Same is true for this. On the minor groove side, we see again 2 hydrogen bond acceptors and 1 hydrogen bond donor. So it turns out that in case of minor grooves, we have less functional groups, plus the gap is also smaller. So small, so there is not much space for amino acid side chains to interact, which means that most of the interactions that we see between protein and DNA are through the major groove. But that does not mean that we do not see any interaction with the minor groove. So it is also there. Similarly, for the AT pair, there is a hydrogen bond acceptor, hydrogen bond donor, hydrogen bond acceptor, and also we have this methyl group which can form very good hydrophobic interactions with hydrophobic residues of amino acid side chains. And this is actually true. So we do see interactions, some of those interactions are shown here. So on top, what we see are the nitrogenous bases, and at the bottom we see various side chains. So what we see here is the interaction of guanine with arginine side chain. So the arginine side chain can form hydrogen bonds with the guanine, and it turns out that they form very nice pairs of hydrogen bonds. So this is the guanidium moiety of arginine side chain, and it can, so this is a hydrogen bond donor, so the H is here and these are the hydrogen bond acceptor. So we can have a different orientation of the side chain. We can have one NH2 group forming hydrogen bonds with both. So there are 2 hydrogens, so each of those can act as a hydrogen bond donor, and similarly shown here. So guanine arginine interaction is very common, and this type of hydrogen bonds are seen quite often. So it means that if you have arginine in the binding site of a protein or a transcription factor, then you can guess correctly that this will most probably form hydrogen bonds with guanine on the DNA in the major groove. Similarly, lysine can form hydrogen bond with guanine. Asparagine can form a pair of hydrogen bonds with adenine. Glutamine can also form a pair of hydrogen bonds with adenine, and glutamine can form a pair of hydrogen bond with guanine. So all these hydrogen bonds are seen quite often in various protein DNA structures that have been solved. This table summarizes some of such interactions. So I am not going to go through all of these. You can pause the video here and look at it for yourself. I will just highlight 1 or 2 interactions. So I said arginine and guanine, right? So this is arginine, and its interaction with all the 4 bases are shown here, and these are all hydrogen bonds. Okay. So for the number of proteins, several proteins were studied here, and arginine side chains were found to interact 24 hydrogen bonds between arginine and thymine, 8 between arginine and cytosine, 19 between arginine and adenine, and 98 between arginine and guanine. So you can see that there is an overwhelming number of hydrogen bonds between arginine and guanine. So this is most probably the most common interactions that is seen. Similarly, if we look at glutamine, you can see that glutamine forms hydrogen bond very efficiently with adenine. So glutamine and asparagine both, so the side chains are very similar, only the distance varies. So glutamine and asparagine both form nice hydrogen bonds with adenine, right? So how does this look structurally? Structurally, it looks something like this, where we have a helix, right? So this type of helices are called DNA recognition helix. So this helix will fit nicely into the major groove. So it turns out that the size of the major groove and the sort of the radius of the helix fits so well that the side chains that are coming out of this helix can nicely interact with the bases as well as the phosphate and sugar group on the DNA. So a helix will insert itself like this and it can interact with the DNA. So it can interact with the sugar and the phosphate group, which will give it non-specific interactions, but of course contribute to the binding affinity. And the side chains, some of the side chains will also interact with the nitrogenous bases, which will give it the specificity towards that particular sequence. So another thing that I would like to point out here is this particular motif called helix turn helix motif. So what are motifs? Motifs are something that are found in proteins, in several proteins. So we have talked about hierarchy of protein structure. So I talked about primary structure, secondary structure, tertiary structure, and then also quaternary structure, right? So secondary structure is where we have these helices and beta strands, and then quaternary structure is where all these secondary structures come together to form the three-dimensional folded form. Motifs are something that is in between secondary structure and tertiary structure. So for example, this one is a helix. So this is one helix, then there is a turn followed by another helix. So this is called a helix turn helix motif. Now this particular motif is found in many structures. So this one by itself will not be able to form a stable structure, right? So this will be a part of a tertiary structure, but this helix turn helix motif is found in many proteins, especially those that bind DNA. And this motif will bind DNA like this, where this second helix of the helix turn helix motif turns out to be the DNA recognition helix, and it will insert itself into the DNA major groove. So we'll see one example. This helix turn helix is found in these two proteins. So one is 434 repressor. So this is like the lac repressor that we have discussed before, and P22 repressor. Now if you see, this is the helix. So the DNA binds this side. Okay, so the residues which are on this side of the helix, they will interact with the DNA, right? And you can see that these residues are different. So in this case, it is valine, here it is glutamine, here it is glutamine, here it is glutamine, right? And then some other residues are different. So depending on the residues which are on this half of the helix, they will interact with the DNA, and those interactions will determine what sequence this helix will recognize, which means that we can engineer these proteins by changing these residues which are on this side of the helix that interact with the DNA. And this is exactly what was done. So a redesigned hybrid 434 repressor was formed, where these wild type residues were replaced by these residues. So you can see that this is changed by this valine, this is here, and then some of other residues like this glutamine is also changed by serine, and right, like so. So now when these changes have been made, so this asparagine is here instead of glutamine. So when these changes were made, so the overall tertiary structure is that of 434 repressor, but only these few amino acids were changed, which are coming from P22 repressor, and now this hybrid structure will bind the DNA for P22 repressor, not that of 434 repressor, right? So we have changed, or this was done. So these residues have been changed, which are towards the DNA, and that changes the specificity of the DNA binding for this particular protein. Okay. So, that concludes our non-allosteric interaction. So, we have seen how small molecules interact with proteins, how proteins interact with proteins, and how DNA interact with proteins, and they all have their unique principles, and we can use those, if we understand those principles, and we can use those principles to modulate these type of interactions according to our need and benefit. The second one that I am going to talk about is allosteric interaction. So, in allosteric interaction, the protein will have two binding sites. In one site, the modulator will bind, and once it binds, it changes the shape of the protein. It can change the whole shape, or it can change only a particular part of the protein, right? So, shape or conformation, and once this change happens, now the substrate can bind. So, allosteric comes from Greek words allos, which means other, and stereos, which means solid or shape. So, allosteric proteins attain other shape. Okay. So, this other shape or conformation upon binding to molecules, which we call modulators. Now, we have already seen some examples. So, we have seen the example of the lac repressor. Allolactose is a modulator which binds lac repressor. It induces a change in the protein so that it cannot bind DNA. So, allolactose will be a negative modulator, right, because it results in unbinding of the DNA, right. So, when it binds to the protein, the substrate cannot bind or the DNA cannot bind. There is another reverse example of this, where cyclic AMP binds to the catabolite activator protein, and it induces a conformational change in its DNA binding, so that now it can bind DNA. Okay. So, this is reverse of the lac repressor and allolactose. So, in this case, cyclic AMP is a positive modulator. So, let us look at hemoglobin, because I am going to talk about allosteric, and hemoglobin is a classical example of a protein which shows not only allosteric but also another important phenomena called cooperativity, right. So, here are two proteins. The one shown on left is myoglobin. It is a single polypeptide chain, and in its binding site it has this heme group, which has a porphyrin ring and a ferrous ion, and this is the one which will bind oxygen. Hemoglobin has the exact same structure as myoglobin. Additionally, it has 4 subunits. Instead of just one polypeptide chain, there are 4 polypeptide chains, and each of them carry this heme group. So, hemoglobin, so each of them can carry this heme group. So, hemoglobin can bind 4 molecules of oxygen. It turns out that myoglobin is actually the first protein whose structure was solved by X-ray crystallography. It is not a very big protein. It has only 153 amino acid and a heme group. Hemoglobin has 4 chains, and their structures are very similar to that of myoglobin, and it turns out that there are 2 alpha chains and 2 beta chains. So, these 2 sequences are same, and these 2 sequences are same. Myoglobin is better suited for storage of oxygen, whereas hemoglobin is better suited for transport of oxygen, and we will see what we mean by that in the next few slides. So, if we compare the structure of myoglobin and one of the subunits, let us say the beta subunit of hemoglobin, then you will see that they look exactly the same. So, we have very similar alpha helices in the same positions, and the heme group also binds in the same site. So, this is the heme group. It consists of 2 parts. One is a porphyrin 9 moiety. So, porphyrin, depending on these groups that are present in these sites, it can be classified as different numbers. So, the one which we are talking about is porphyrin 9, and there is a ferrous ion at the center, and this ferrous ion forms an octahedral geometry. So, it coordinates with these 4 nitrogens. Then at the bottom there is this proximal histidine. So, this histidine is on helix F. It also forms this coordination. So, this will be a nitrogen atom on the histidine, and the 6th coordination is formed by the bound oxygen, and there is also another histidine which is called the distal histidine, which is on the E helix of this protein. So, together this forms the binding site of the protein. Now, there is something that is very interesting. So, oxygen is something that hemoglobin carries from our lungs to our tissues. It turns out that carbon monoxide, which will have very similar structure and shape as oxygen, it binds to the heme group 200 times tighter than oxygen, right? And this is something that is very problematic, because carbon monoxide, if it binds this, then it acts as an inhibitor, right, of the heme group. So, it will prevent the hemoglobin to bind further oxygen, which means that the number of available hemoglobin molecules will decrease if there is carbon monoxide present. So, that will result in lower supply of oxygen, and that is carbon monoxide poisoning, and it is very dangerous because carbon monoxide is a colorless and odorless gas. So, in less ventilated places where you have some source of carbon dioxide, maybe from some fire or maybe from some diesel generator that is running, it can create substantial amount of carbon monoxide that the person can breathe, and that will result in inhibition of hemoglobin. So, let us look at some facts about hemoglobin. It turns out that the solubility of oxygen is very low in aqueous solutions. So, it is around 7.7 milligram per liter in water at 35 degrees centigrade, very close to our body temperature, and 1 atmospheric pressure, right. So, which means that since we need to carry oxygen in large distances from our lungs to our different body parts, we need to increase the solubility, and that is where hemoglobin comes in. Red blood cells, which are also called erythrocytes, they carry hemoglobin in such a way that 1/3 of these cells, one-third weight of these cells are actually coming from hemoglobin. So, they are tightly packed with hemoglobin, and these cells, they survive roughly for 4 months, 120 days. Erythrocytes are very interesting cells. We will discuss cells in more details in the next week's lecture, but I just wanted to point out a few important facts or interesting facts about these red blood cells. So, they are formed from precursor stem cells called hemocytoblasts, and then once this is formed, of course, they will need all the cell organelles to produce hemoglobin. So, they produce large amount of hemoglobin, and once that is done, then they do not need the DNA, they do not need all the other machineries which are needed for protein production. So, all of these organelles are extruded out, right. So, they are all thrown out, like nucleus, mitochondria, endoplasmic reticulum, these are all thrown out, and the cell is only packed with or majorly packed with hemoglobin. This is something that will be important, and we will see this again and again in the next few slides. Partial pressure of oxygen in our lungs is 13.3 kilopascal, and in our tissues is 4 kilopascal. So, it is of course less partial pressure in tissues compared to the lungs. Now, we will see curves like this. So, theta is a term which is saturation, right. So, saturation means that theta has a value of 0 to 1. So, if it is 0, it means that hemoglobin has no oxygen bound to it. If it is 1, it means that hemoglobin is completely saturated with oxygen, and we can express theta in terms of this. Now, here the expression is written in such a way that we are assuming that one molecule of hemoglobin binds only one molecule of oxygen. So, this will be more for myoglobin, not hemoglobin. We will have to modify this expression for hemoglobin, right. So, concentration of oxygen or the ligand divided by concentration of the ligand plus Kd, right, and we can replace this concentration of the ligand with the partial pressure of oxygen, and we can replace Kd with P50, which is the partial pressure at which 50% of the molecules are bound to oxygen, or theta equals to 0.5, right. Now, if we have this, if my Kd or P50 is low, then my binding curve will look something like this, and if my Kd or P50 is high, then it will look like this, right. Now, this is where this is the partial pressure of oxygen in lungs, and this is the partial pressure in tissues. If the affinity is low, then our theta will be somewhere here. So, let us say 0.5, So 0.5 or 50% of myoglobin like molecule will bind oxygen, and then when it goes to tissues it will be around 20%. So 50 minus 20, only 30% of myoglobin will release oxygen, right. So the release of oxygen that we will get is only 30%. On the other hand, if it is binding very tightly, let us say this is 99% and this is 98%, then the release of oxygen is only 1%, even though it is binding oxygen, but it is not releasing it, right. So that is why myoglobin is a very poor oxygen carrier, right. We do not want that. We want something different, and that is where hemoglobin comes in. Its binding curve does not look like this, which is a hyperbola, right. It looks like this sigmoid curve, right. So in the lungs, it binds oxygen, which is, let us say, around 98% saturation, and in the tissues it comes down to, let us say, 50%, right. So 98 minus 50, that is 48% of unloading of oxygen, right. So almost 50% of, as if 50% of hemoglobins are carrying oxygen between lungs and tissues, right. So this is much more efficient than these two. So how do we get that? This results from a particular phenomenon called cooperative binding of ligand, and cooperative binding of ligand arises when there is allosteric between multiple subunits of a protein. So just allosteric in a single polypeptide chain will not work. You need multiple subunits and you need allosteric between them so that there is cooperative binding, and when all of these things happen together, we get a sigmoidal response like this. So let us look at the hemoglobin structure again, and it turns out that hemoglobin actually has two different conformations, right. So this is one conformation which is referred to as the T state or the tense state, and this is another conformation which is referred to as the R state or the relaxed state. Hemoglobin has lower affinity for oxygen in the T state and higher affinity of oxygen in the R state. Also, oxygen binding shifts the equilibrium from the T state to the R state. So what is the difference between these two? There are all these different ion pairs which are, or these are actually electrostatic interactions between different amino acid side chains. So these are present in the T state which are not there in the R state. Also you can see that this central portion is different, right. So structurally they are quite different, significantly different from each other. So how do we get this structural difference? It turns out that it is triggered by the binding of oxygen to the heme group. So in the absence of oxygen, so this is the deoxyhemoglobin, the porphyrin ring looks like this. It has a slightly bent structure, right, and then this proximal histidine is interacting to the iron. So in this case the iron is in a high spin state. When oxygen binds, it changes the iron to the low spin state, right, and this heme group flattens. So you see it is slightly bent, here it flattens, and that results in a tug on the proximal histidine which rotates, and this small change results in a change in the helix F, which results in bigger changes in the rest of the protein. So all these ion pairs are changed and the protein actually changes its conformation, right. Another important interesting fact happens that it goes from T state to R state. So the neighboring subunits also go from T state to R state, right, which means that they can now bind oxygen more tightly, right. And this is where cooperativity comes in, that once one molecule of oxygen binds to a particular subunit, the next oxygen will bind tighter, the third one will bind even tighter, and the fourth one will bind even tighter, right. And that is why we see this sigmoidal curve where it zooms up as more and more oxygen binds, and then of course it reaches saturation. So to understand that we have to plot, and the cooperativity can be plotted nicely by something called a hill plot, right. So before I do that, let's introduce two more terms in terms of allostery and cooperativity. So we saw that in case of lac repressor and cap protein, that there is a modulator which results in the change in DNA binding affinity. So it is either allolactose for lac repressor or it is cyclic AMP for cap, right. So here the modulator is different from the ligand, and this type of interaction is called heterotropic interaction. But in case of hemoglobin, the modulator and the ligand are both oxygen. So binding of oxygen to one subunit results in tighter binding of oxygen to the second subunit, right? So here the ligand and the allosteric modulator are same. In this case, the interaction is called a homotropic interaction. And we can represent binding of hemoglobin to multiple oxygen molecules like this. So in this case, N will be 4, right? So hemoglobin plus 4 molecules of oxygen gives you hemoglobin oxygen 4, so O2-4 complex, right? You can also divide it into 4 equations. So hemoglobin binds one oxygen, so it will be Pl, then another oxygen it will be Pl2, then another oxygen it will be Pl3, then another oxygen it will be Pl4. So if we do that, our Kd gets modified like this, where instead of having just L, we have L to the power N, because we have N number of ligands here. So it will be P times L times L times L times L. So L repeated 4 times. And then this will also modulate, change our saturation equation like this, where we replace L with L to the power N, and this is Kd. So we can replace. So now if I subtract 1 from this and then divide it by 1 minus theta, I can change this equation to look something like this. So theta by 1 minus theta equals to L to the power N divided by Kd. If I take log on both sides, then left hand side will be log of theta by 1 minus theta, right hand side will be log of this. So it means log of L to the power N. So N comes in the front, so N log L minus log Kd, right? Now what we can do is we can replace this with partial pressure of oxygen, and we can replace this with P50. So if we do that, then the equation looks like this, and again Kd will be P50 to the power N. So we get a N in the front, right? So now if I plot log of this versus the partial pressure of oxygen, this is a straight line equation, right? And remember that it's always easier to extrapolate a straight line data, right? So that's why people will always linearize equations like this. So if we plot this versus the partial pressure of oxygen, it will look something like this. So what is this? Now you see, I mentioned that hemoglobin has two different conformations, T state and R state. In T state, it binds oxygen weakly. In R state, it binds oxygen tightly. So T state is the one which is there. Binding of oxygen will convert it slowly towards the R state, right? So this is my log theta by 1 minus theta. So binding of oxygen, and this is my partial pressure of oxygen, and both are in the log, right? So at lower concentration of oxygen, hemoglobin is in the T state. So we will get a curve like this. So this straight line represents the binding of oxygen to the T state. When the partial pressure is more, then the affinity increases. So it shifts, and this straight line represents binding of hemoglobin or binding of oxygen to hemoglobin in the R state, right? But then what actually happens is initially most all the hemoglobin are in the T state and oxygen is binding. So it will follow this curve, and then as more and more hemoglobin or more subunits get converted to the R state, it will depart from this and it will start moving towards this affinity, right? Once it reaches this, then of course it will follow this line. So we have two different slopes. This slope is this, which is 1, and then there is this slope which has a Hill coefficient of 3, right? So this Hill coefficient of more than 1 represents cooperativity, okay, which means that once oxygen starts binding, it will shift the equilibrium towards the R state, which means that the next molecules of oxygen will bind hemoglobin with tighter affinity, right? So in plots like this, whenever we get a slope that is more than 1, we say that there is cooperativity between different subunits. So how do we explain that? There are two models that were proposed in the 1960s, right? So one is called the concerted model, and the other one is called the sequential model. And these were proposed by two groups of scientists. So the concerted model was proposed by Jacques Monod, Jeffrey Wyman, Jean-Pierre Changeux in 1965. Next year, Daniel Kostland and his group members and his co-workers, they proposed the sequential model. Right? So let's look at this. The concerted model states that all the subunits are either in the T state, which binds oxygen weakly, or in the R state, which binds oxygen tightly. Right? And then we have equilibrium between all these different states. So you can see there are Kd values here and there are equilibrium constants here. Right? So as more and more oxygen binds, the equilibrium will be shifted towards this right hand side. So less molecules of hemoglobin will be in this state and more molecules of hemoglobin will be in this state. So in the free state, all will be in the T state, and when we reach saturation, all of them will be in the R state. Right? Sequential model says that all the subunits can be either in the T state or in the R state. Right? So the number of possibilities are of course much more. So again, in the free state, all of them are in the T, and when all the subunits are bound to oxygen, it will completely shift towards R, and there are all these different multiple paths by which it can start from here and reach here. But overall what happens is, as this T gets converted to R, your affinity will keep on increasing. So the equilibrium will keep on shifting from this left hand side towards the right hand side. Right? So there is more to oxygen binding to hemoglobin. It turns out that hemoglobin also carries back carbon dioxide from the tissues to the lungs. So in the tissues, carbon dioxide is formed and it can react with water to form this bicarbonate ion and protons, right? And this reaction is actually catalyzed by an enzyme called carbonic anhydrase, and this carbonic anhydrase is present in a lot of concentration in the erythrocytes or the red blood cells. So hemoglobin transports 40% of proton and 20% of carbon dioxide produced in the tissues to the lungs. So it is important to note that solubility of carbon dioxide is much more in water than that of oxygen. So the remainder of the proton that is produced is absorbed as bicarbonate in blood plasma, and it is carried through that. Remainder of carbon dioxide is transported as this bicarbonate. So 70% of it will be like this, and 20% will be and 7% will be dissolved carbon dioxide. Okay. So 20% is carried by hemoglobin. Now, because of this production of this hydrogen ion, the pH in the tissues is actually lower than that in the lungs, 7.2 versus 7.6. And it turns out that this lower pH and high concentration of carbon dioxide decreases oxygen affinity of hemoglobin in the tissues compared to that in the lungs. That is what I am saying here. So this influence of pH and carbon dioxide on hemoglobin binding to oxygen is called Bohr effect. So Bohr effect was first described by Christian Bohr in 1904, and he turns out to be the father of the famous physicist Niels Bohr. Okay. So how does hemoglobin carry carbon dioxide? So carbon dioxide is a much bigger molecule. It is not going to bind the heme group. It actually binds or reacts with the amino terminal of the polypeptide chains. So this is the free NH2 group of the polypeptide chain, the N terminus. Carbon dioxide can react with it to form this type of carbamino terminal. Okay. So now this results in the changing of this ion pair interactions. So it actually stabilizes the T state, right? So if it stabilizes the T state, what happens? Affinity of oxygen binding will decrease. So let's look at this. This is the pH dependence of hemoglobin. So this is the same plot, percent saturation, you can think of it as theta, partial pressure of oxygen. So in lungs, this is the partial pressure, and in the tissues, this is the partial pressure. In lungs, the pH is 7.6. So the curve will look something like this, the one on the extreme left. As pH decreases from lungs to tissues, the curve will shift. So, it will be this right hand one in the muscle, right. So, in the lungs, the pH is high, pressure is high. So, this will be the saturation. Okay. So, almost 100%, let us say 98 or 99%. In the tissues, the partial pressure has decreased, and because of pH and carbon dioxide, this has also shifted. So, now it is here. So, you can see that 98 minus 30, so that is almost 70% of oxygen offloading happens between the lungs and the muscles because of this effect, because of the Bohr effect. So, there is another molecule which is called 2,3-biphosphoglycerate or BPG, which also modulates the binding of oxygen to hemoglobin. So, it turns out that BPG interacts with the two beta subunits like this. So, you have these negative charges. So, these negative charges will form ion pair interactions with the positive side chains of the beta subunits, and it also stabilizes the T state as compared to the R state, which means that the presence of BPG reduces the oxygen affinity of hemoglobin. So, what is the effect of that? Effect of this is seen when we are in a condition of low oxygen availability or low partial pressure of oxygen. So, one example will be that if you go for mountain climbing where the partial pressure of oxygen is less. So, this is the partial pressure of oxygen at sea level. This is the partial pressure of oxygen when you are at a height of 4,500 meters, right, and this is the partial pressure in the tissues, which remains the same. BPG, it turns out that amount of BPG is changed between these two conditions, right. So, when there is less BPG, oxygen binds by this curve which is shown on the left, right. So, we have less BPG under normal conditions. So, oxygen binds here and then it releases in the tissues. So, you will see these numbers are changing between plots, but the idea is the same. So, let us say 38% of oxygen is released. If the same curve persists, then at a height the partial pressure has decreased, so it binds here, and then the tissue is the same, so it releases here. So, this is only 30%, right. So, instead of 38%, only 30% of oxygen is released, which means that available oxygen will be less. But if the curve shifts to this one on the right hand side because of more concentration of BPG, then less amount of hemoglobin will bind oxygen, but the release will be greater, and you can see that this number is 37%, which is almost the same as this. So, even though the hemoglobin is less saturated, but the offloading is the same, which means that the oxygen availability becomes almost the same due to the increased concentration of BPG, right. Okay. So, you can read about all of these in these different books. So, I have suggested several books. You can go through them and read the details about protein ligand interactions and hemoglobin and allosteric. Thank you.